we're going to start with just a quick review of microbiology as a whole. If you've had microbiology class before, if you remember something from a general biology course, even maybe some human anatomy, physiology, some of this stuff might be a review for you, so I'm hoping that a lot of you guys already know some of this stuff, and this will just be a really quick refresher. First of all, when we talk about bacteria, which is what we're talking about in this section, we're just talking about bacteria. Bacteria are what we call prokaryotes. They're prokaryotic cells. And what that means is their cells are very, very simple. They're not going to have all kinds of organelles inside of their cells. They're not going to have a membrane-bound nucleus. They're going to be very, very simple. On the contrary, more complex cells are eukaryotic cells, or eukaryotes. We as humans are eukaryotes. Plants are eukaryotes. Fungi are eukaryotes. So your more complex organisms are going to be eukaryotic. So we're going to talk about the bacteria, which are prokaryotes. Very, very simple organisms. And the bacteria are also unicellular. What that means is they are composed of one cell. One cell, that's it. So one bacterium is just going to be one cell. It's unicellular. We as humans, we're multicellular. We're made up of lots and lots of different cells. So it's not just one cell that makes us up. So with bacteria, they're unicellular. They're going to be very simple. Now, a few pieces of cell structure to talk about. One of the ones that's most important with bacteria is going to be the cell wall. And the bacterial cell wall is composed of this component that's called peptidoglycan. And what that is it's going to be these sugar and protein linkages. And they are going to build the cell wall of the cell, and they're going to surround it. They're joined together kind of like a chain link fence. So they are going to surround the cell. They're going to give that cell structure. They're going to give it support. And they're also going to give that cell its shape. They're going to keep that cell from bursting. If a lot of water comes into the cell, they're going to hold its structure up to a point. Cells can always burst, but they're going to give it its structure up to a point. So they're going to maintain stability, maintain structure, help keep the cell from bursting or lysing. Lysis is another word that we use when cells burst. Now, one other thing that's important about that peptidoglycan and that cell wall is it allows us to differentiate between two different types of bacteria. And this is a very important concept, and this is something that we're going to encounter in this class over and over. But what that peptidoglycan does is it lets us differentiate between your gram-positive cells and your gram-negative cells. So those are two different types of cells, and they're going to be divided based on their cell walls. Now, when you use the word gram, it's always capitalized. The G is always capitalized because it's named after a scientist. He invented the gram stain. I'm going to put up a little video about the gram stain. It kind of shows you how to do it. But a gram stain will differentiate between gram-positive and gram-negative. But with gram-positive cells, these guys are going to have a really, really, really thick layer of peptidoglycan. So they're going to have lots of peptidoglycan that makes up their cell wall, a very, very thick layer. With your gram-negative cells, they're going to have a very thin layer of peptidoglycan, but what they're going to have in addition to that is they're going to have an outer membrane. So they're going to have a thin layer of peptidoglycan, and then they're going to have an outer membrane. Now that outer membrane, one of the things that composes that outer membrane, it's a bilayer, which means it's two layers, but there's going to be a lipopolysaccharide layer, and nicely enough we call that LPS. We shorten that LPS. So it's an LPS component of that outer membrane. Now, that's going to be really important because one of the things that makes up that LPS layer is a guy who's called lipid A. And lipid A can actually act as a toxin. So that's going to be important when we talk about your gram-negative bacteria. So gram-positive, very thick layer of peptidoglycan. Gram-negative, thin layer of peptidoglycan, but then there's going to be an additional outer membrane, or that LPS layer. And that LPS layer does contain lipid A, which is something that we'll talk about in the future. So moving right along, I got cheat notes so I don't get too far off track. A couple other parts of the cell to mention while we're talking about that. We briefly talked about the cell wall, and you have notes about this in Blackboard. Cell wall, also the cytoplasmic membrane, depending on what textbook you've learned from, what teacher you've learned from, that can also be called the plasma membrane or the cell membrane. I typically use cytoplasmic membrane. This guy, again, he's a bilayer. He's made up of 2 layers. Cytoplasmic membrane is very, very important because he's going to regulate what comes in and out of the cells. You don't want just anything coming into the cells. You don't just want anything going out of the cells. So that's very, very important. He's going to be a regulator. And because bacteria cells are prokaryotes, and I mentioned they don't have all these organelles to do jobs, your cytoplasmic membrane is going to do a lot of those jobs. It's going to be where the cell's going to generate energy. Your proteins are going to be synthesized there. Like I said, it's going to regulate what comes in and out. So lots of jobs for your cytoplasmic membrane. So it's going to be very, very important in your bacterial cells. A couple other things. Let's see. Ribosomes, that's where your proteins are built. Proteins are important for enzymes. It's going to be important for structure. Nucleoid, that's where the cell's DNA is contained. Cells are just going to have one circular chromosome of DNA in their nucleoid. Now there is another part of DNA that cells can have, and this is completely separate from their chromosome. So their chromosome is going to be their absolute crucial part of DNA. But what cells can also have is they can have what are called plasmids. Now these plasmids are extrachromosomal DNA. What I mean by extrachromosomal is they're aside from the chromosome. So there's the chromosome that they have to have, and then there's plasmids which are extrachromosomal. Now these guys can hold information that can be beneficial to the cell, but not necessary for regular life. So, for instance, plasmids can contain information for what we call virulence factors. Those are things that are going to make the cells more virulent. Things like the cell's pili and flagella, which those can help with adhesion, which the pili can, or flagella, that can help them be transported, help them move. They can move like in a whip-like motion that can move the cells. Things like capsules and slime layers. These are things that will go outside of the cell. Capsules can help them, or both capsules and slime layers can help the cells kind of be cloaked to help them evade detection from the immune system. So they can help them be more pathogenic. Slime layers can also, they can help the cells adhere to different locations where they want to be. So your plasmids, like I said, it's extrachromosomal. Some toxins are on plasmids. It's extrachromosomal. It's not part of the chromosome. It's separate. These are things that can give the cell advantage. They can give them advantages, but they're not necessary for life. So that's what plasmids are. They're extra information. So they carry extra DNA. And also, the bacterial cells can transfer those from one guy to another. So plasmids can be transferred, which can be kind of a scary and kind of a bad thing, because if they have a plasmid that codes for a toxin or a plasmid that codes for some other virulence factor, they can share that. So they can share that with other bacteria. So those plasmids can be shared. So that is a very important concept to note on. Let's see, a couple of well, let's see. That's pretty much what we talk about with your cell structure. That's why I have notes, because I'll just keep going on. Next thing that we hit on in the notes is microbial growth. Your microbes are going to reproduce by what we call binary fission. What happens is there's a cell. Start with one cell. That cell will completely replicate itself, form a septum or a wall, and divide off. And it'll start with the parent cell, and you end with a daughter cell. So it's going to completely replicate itself. It's going to be an identical copy of itself. The only time that they're not identical copies are when problems with DNA transfer come about, and then you're going to get different guys. But ideally, they're going to just make complete carbon copies of themselves. Now, bacteria grow by what we call exponential or logarithmic growth. Now, what that is kind of, we already started to touch on it. You have one cell, and then that cell's going to divide. So you have one cell that ends up in 2 cells. Now what will happen is those 2 cells will then divide. Those 2 cells will go to 4 cells. 4 cells will divide into 8 cells. 8 cells into 16. 16 into 32. 32 into 64. And so on and so forth. So what happens with this growth, we call it logarithmic or exponential growth, is with every generation the number of cells doubles, which can be kind of concerning, especially if these guys are in your body, if they're in your food. And the time that it takes for them to double is called their generation time, or their doubling time. So their generation time or their doubling time. It's the time that it takes them to double. For instance, E. coli has a doubling time or a generation time of around about 20 minutes. So every 20 minutes, the entire population of that bacteria will double. And that's assuming that they're under ideal circumstances, that they have the nutrients that they need, oxygen requirements, and everything like that. So that's an important concept. So every time that these guys, their doubling time happens, the whole number is going to double. So that's going to be your exponential growth or your logarithmic growth. Lets them get to really high numbers really, really, really quickly. Let's see, a couple other things. The stages of microbial growth. If you put bacteria into like a closed system, if you're growing them in the lab, put them in a test tube, put them on a dish, what you're going to see are these 4 distinct patterns of growth. Now these happen if they're in our food, if they're in our bodies, but we study them, you know, in a lab setting. That's how we get this information. But for our 4 stages of growth, we have 4 stages. We have lag, log, stationary, and death. Now the lag phase, there's not a lot of increase in cell number. Cells are priming. They're getting ready to replicate. They're getting ready to reproduce. So they're starting to generate the ATP that they're going to need. They're starting to build up the molecules that they're going to need in order to reproduce. So they're just getting ready. So it's not going to, you're not going to see a real increase in cell number then. They're priming. It's kind of a priming stage. So that's your lag phase. Log phase, that's when you have exponential growth. That's when these cells are doubling with every generation according to their doubling time. That's when they're going crazy. They're going nuts. They're so happy during the exponential phase. So and the lag phase and exponential phase, those can be used interchangeably. Typically we just say lag phase. I'm sorry, log phase. Lag is when they're priming, log is when they're going nuts and they're going crazy and they're growing like crazy. So in the log phase or the exponential phase, that's when they're growing, they're replicating like crazy. Next phase is stationary phase, and the lag phase is going to continue. The log phase, guys, start with the lag phase, and then you go to the log phase. It's even alphabetical. That's how I always remembered it. But in the log phase, when they're having this exponential growth, that's going to keep on until they start to run out of nutrients. They're going to build up waste. They're going to run out of nutrients. So things aren't going to be ideal forever for them. So when that happens, they're going to transition to the stationary phase. Now, in the stationary phase, if you see it on a graph, and there's a link to a graph to look at, it's going to be a plateau. What happens there is you're still going to have cells that are finishing their replication cycles, and then you're also going to have cells that are dying. So there are going to be new cells produced, and there are going to be old cells dying. But what happens in the stationary phase is that the overall number of cells is going to stay about the same. So you've got some that are finishing replication, some that are starting to die. So your number is going to be pretty even across the board for your stationary phase. And the last phase is death phase, which makes sense. They're going to die off. And they're going to die off slower than they grew. So in that log phase where they grew like crazy, on a graph you'll see that as being really just a steep jump. And then the death phase, they're going to taper off a bit more slowly. One thing that I mentioned in the notes is that when we talk about bacterial growth, we talk about cells either multiplying or dividing. And you know, in a mathematical term, in just normal conversation, they don't always mean the same thing. So just be careful with when you're thinking about it with bacteria, because you can read in any textbook. Some textbooks will say that the cells are dividing, or some will say that cells are multiplying. Really what we think about is the number of cells is multiplying. You're getting more and more and more cells, and we think of multiplication as another word for reproduction. And then when we say the cells are dividing, what we mean is literally, the cells are literally dividing. So when we say that these cells are dividing, we're not talking about a mathematical term for dividing. We're talking about a literal cell division. So sometimes you'll come across that in reading, and you'll see that the cells have divided. What it means is not the cell number has divided, not the total number of them. It's not getting any smaller, but the cells are literally dividing, and the number is multiplying. The number of cells is multiplying, because again, that's your exponential growth. Get to really high numbers really fast. And let's see, a couple other things, just real quick. We end this section talking about some of the pathogenic features of bacteria. One of them, I'm actually going to do these in a little bit different order than it's in your notes. I'm going to start, let's see, you talk about endotoxins first, talk about endotoxins. If you recall, I spoke about lipid A. lipid A being part of that lipopolysaccharide layer in your gram-negative bacteria. Lipid A is an endotoxin. So that happens with your gram-negative bacteria. So they can have an endotoxin. In fact, all gram-negative bacteria possesses endotoxin. Usually during cell multiplication or cell death is when that endotoxin is going to be released. And they're going to cause a stimulation in the host immune system. They can even cause you to go into shock if a lot of it's released all at once. So your gram-negative bacteria, all those guys are going to have endotoxins. Now, exotoxins are a soluble protein. It's going to be released by either gram-positive or gram-negative bacteria. So exotoxins can be either gram-positive or gram-negative. And these guys can destroy cells. They can mess up with the metabolism. The cell can release them as the cell's alive, or they can release them as it's dying. And these guys are going to be more potent than your endotoxins. A couple of examples. We talk a lot about enterotoxins with foodborne bacteria, and those are going to just wreak havoc on your gut. So we talk a lot about enterotoxins with that. Also, a neurotoxin, botulinum, Clostridium botulinum. He secretes a neurotoxin, it's botoxin, and that's going to kill neurons or your nerve cells. And it's also the same guy that we get botulism from, or I'm sorry, you get Botox from. Botox is botoxin, so also called botulism. But it's a neurotoxin, and that's a type of an exotoxin. And one other pathogenic feature of bacteria, and these are typically with your gram-positive bacteria, is the formation of endospores. And not all guys are going to do this. It's typically gram-positive bacteria. But when cells do form endospores, it is notable. So you'll find when we study about cells, we'll say if they form endospores or if they do not. But what happens is they will sporulate and they'll form an endospores. Endospores are dormant. They're just hanging out. It's not a form of replication, anything like that, but what they're doing is they're just hanging out. This will happen when conditions are not favorable. So when there's not a lot of nutrients available, when just temperatures aren't great, the environment's not great, there's not a lot of water. Whenever environmental conditions are not favorable, these guys who produce endospores will sporulate. They'll do that. And these endospores can survive those unfavorable conditions. They just hang out and just waits for things to get better. And when things do get better, then that endospore will germinate. It'll become active again. It'll become an active cell. So they'll germinate and just be a regular active cell again. But that's very important because it can allow these guys to survive pretty harsh conditions. And one of the guys who does that, one of the big guys who does that, are your Clostridiums do that. Also your Bacillus species. But sometimes you'll hear about bot spores, botulism spores. Anthrax is another guy. You'll hear about anthrax spores. But anyway, that's what happens with sporulation. These guys are going to be formed when conditions are not favorable, and it's just going to be a way that allows them to get through these unfavorable times, and then when things get better, then they can germinate, they can become active once again. So that's just another factor that increases the pathogenicity, and of course being able to produce a toxin, or with your gram-negative bacteria, having a toxin kind of already built in that LPS layer. Those are also going to increase the pathogenicity of the bacteria. So that kind of sums up what your notes talk about in this section. So if you have any questions, just let me know. Thanks.